Hello, my name is Martí from the Ph.D. Parsons Group in Edinburgh, and I'm gonna show some results from a study where we investigated the synaptic localization of TMM97 and its relation with amyloid beta in human Alzheimer's disease cases. Briefly, as an introduction, it is well known that synaptic loss is the best correlate with the cognitive decline that we found in Alzheimer's disease, as for example, in this article where lower synaptic cones were related with lower mental states. But the mechanism by which we have this synaptic loss that is believed to occur before the massive neuronal degeneration are less clear. There have been proposed many mechanisms. We have, for example, the extracellular aggregation of the characteristic aggregates of beta amyloid in form of plaques, the intracellular aggregation of hyperphosphorylated tau, but also other players such as microglia. Among those, the usual suspects that have been since the beginning of Alzheimer's disease, the aggregation of beta amyloid is still in the center of the attention. Nowadays, mainly from in vitro and biochemical studies, it's believed that maybe not these big beta amyloid plaques, but oligomeric forms or small aggregates of beta amyloid are the ones that are closely related to this synaptic degeneration. In human cases studies, the evidences point towards an accumulation of beta amyloid inside the synapses. For example, in the first study done in human cases, led by Prof. Parsons, it was shown that in postsynaptic terminals, in this case labeled by Ph.D. 95, we could find beta amyloid, here labeled with NAP61, close to the core of the plaque. So this is the distance from the core of the plaque, and we can see that we have more synapses with beta amyloid. Also, this was related with a reduction of the postsynaptic density cones in the vicinity of these beta amyloid plaques. So we see that we have these small aggregates or oligomeric forms inside the postsynaptic terminals, but we don't really know what is doing and how it's entering. That's why there is also a huge field of investigation that tries to find binding partners of beta amyloid in the synapses. Here you can see a list of some of the ones that have been described until now. Most of these binding studies come from biochemical studies or animal model works, and this is because the interaction is a functional thing, and because we don't have many tools to study that in human brain. I have to say also that probably the most replicated one and then the most accepted is the cellular form of prion protein, and you will see that we have included it also in the present study. But what we wanted to focus is in the role of TMM97. Why? So first of all, TMM97 have been linked by different ways to Alzheimer's disease. It has been shown that if we use antagonists of TMM97 or the sigma-2 complex, receptor complex, we can see a reduction in the uptake, in this case, in cellular model. Here we have different antagonists, and we see a reduction of the uptake of A-beta for those primary rats cell cultures in this case. Also, it has been shown in other cellular models by his own colleagues, and they have also shown that if we use a mouse model of AD and we treat them with this antagonist, we can see an improvement of the cognitive impairment that is characteristic from AD. Even more, we recently found in a proteomic study where we isolated synapses from Alzheimer's disease cases that TMM97 was increased in Alzheimer's disease cases when compared with cognitively healthy controls. So taken together, we know that we have this amyloid in the synapses. We know that we have increased TMM97 in those synapses, and that by blocking TMM97, we can change the relation of A-beta with the internal levels of beta amyloid. So that's why we wanted to answer the following objective. First of all, we wanted to visualize the synaptic localization of TMM97 in human Alzheimer's disease cases. Second, if we found it, study the relation, the close proximity with amyloid beta that we know that is present in synapses. And finally, if there is this potential interaction, we will try to modulate it and see which effect it has in an animal model. So I will explain the method in each objective. So first of all, to visualize the synaptic localization of TMM97, we used an approach that we use a lot in our lab, that is array tomography microscopy. That's briefly what is based on is in collecting fresh tissue, in this case from temporal cortex. We use an acrylic resin to polymerize it, and then we section it in consecutive sections of 70 nanometers thick sections. It means that we will have a resolution, at least axial resolution, that is below the limit or the diffraction limit of light. So when we reconstruct that, we will have these three-dimensional single synaptic terminals resolved. We will be looking at postsynaptic terminals labeled with Ph.D. then the protein of interest, beta-amyloid, and TMM97. To have an idea of how many cones we have around 1 million single synaptic terminals studied in the present work. So the first thing was to see if we could visualize TMM97. Here you have a maximum density projection of 10 consecutive 70 nanometers thick sections. We have a control case, an Alzheimer's disease case, where we can see the beta-amyloid plaque. We can see this pattern of TMM97 that is quite similar between controls and AD cases, and we can see the Ph.D. and the merge. When we quantify those objects, what we can see already is that there is an increase in Alzheimer's disease cases when compared to controls. Here you can see in blue the Alzheimer's disease cases, and we can see this increase in overall TMM levels. What we did also is to see if there was a relation with the abundance of beta-amyloid. Here we have a core of one of these dense core plaques, and we quantified every 10 micrometers the abundance, the density of TMM97. As you can see here, there are no differences. You can see in gray what we found in control, the mean and the standard deviation, and we can see that the overall levels are increased, but there is no difference as we are close to the beta-amyloid plaque. As previously described, if we look to Ph.D. we'll find a slightly decrease in overall, but not significant, but we will find a reduction close to the beta-amyloid plaque, as before I show you in this Coffey and colleagues article. Also, as expected, we found much more beta-amyloid in Alzheimer's disease cases and much more close to the core of these beta-amyloid plaques. So once we know that we can detect TMM97, we can move to the next step and see how it is related with the synapses. So here you have a 3D representation of one of those Alzheimer's disease case and the three conditions that are quantified below. So first of all, we wanted to know TMM97 if it's present in those synapses. So here you have 4 consecutive 70 nanometers thick sections of these thresholded already images, what we quantify. And here a reconstruction of one of those synapses that you see the postsynaptic terminal in cyan, and you can see TMM97 in magenta, and the white areas in all the figures is the overlap. So if we quantify that, if we quantify those synapses, those postsynaptic terminals that have TMM97, what we see is that Alzheimer's disease cases is much more common to find those synapses. So at this point, more or less, we are more or less replicating what we found already in proteomic studies. So we can say that it's much more common to find TMM97 in synapses of Alzheimer's disease cases than in controls. Then, of course, we found more synapses with beta-amyloid, that is the condition C. And if we think that they may be binding, that TMM97 may be a binding partner of beta-amyloid, we need to look also at the synapses that have both TMM97 and beta-amyloid. And this is the condition D. This is one of those synapses, and we can see that we can find it, and it's much more common to find it in Alzheimer's disease. So at this point, we can move one step forward and study this potential interaction between TMM97 and beta-amyloid. And to do that, what we will use is the Foster resonance energy transfer, the FRET approach. In this technique, we will look for the distance between the fluorophores, in this case, that are labeling either beta-amyloid, that will be the donor, or TMM97, that will be the acceptor. So usually, if they are not close enough, if we excite the donor fluorophore and we record both the donor and the acceptor, we will see only emission of the donor. But if they are close enough, that it means around 10 nanometers, we are not going to calculate exactly the distance. We will see some signal in the acceptor emission under the donor excitation. To do that, we use this pipeline that I will not go through, but it's just to show you that usually when we acquire pictures, we use the excitation and the emission of the different channels, 488 for Ph.D, CY3 for A beta, and CY5 for TMM97, that it will lead to what I've shown before, these different channels that gave us the density, the plaque distance, and the focalization. But for FRED, we will use this excitation of the donor, in this case CY3 labeling A beta, and the emission of the acceptor CY5 labeling TMM97. Of course, those images need to be corrected for direct excitation and crosstalk, but what we end up analyzing and what we decided to do was to do a single pixel analysis where when we found in the same pixel TMM97 and beta amyloid in the synapses, we will look to the percent of those pixels where we see a FRED signal. So what we found is the following. When we calculate the percent of pixels where we found FRED, we saw that between A beta and TMM, around 40% of those pixels we found some FRED. So it indicates a potential interaction, a close proximity. You can see in green here is the technical control. So here it will be the negative control, where we only labeled with donor or acceptor antibodies, and a positive control where we are using both fluorophores labeling beta amyloid. So at this point, we can see that it's inside this range. Although that we wanted some biological also controls, and that's why we included this panel of couples of protein interactors. We included beta amyloid and the prion, the cellular prion protein, as I said before in the introduction, the most replicated one, and you can see that we have similar levels. We included PGRMC1 and TMM97 that have been described to be part of a complex, so it's also inside here. PSD95 and beta amyloid that have been also described that at least in some synapses be binding, and all those were statistically significant, higher than synaptophysin and PSD95, which we know that they are really close, but they are separated by the synaptic cleft, so we were not expecting to find FRED. And we also included PSD95 and PGRMC1 where we did not find a FRED signal. So at this point, we think that, well, we've seen that beta amyloid and TMM97 can be in the same synapses and that they are close enough to be generating this FRED signal that may indicate a close proximity and maybe an interaction. So that's why we moved to animal models to study the effect of modulating this potential TMM beta amyloid interaction. To do that, we use an AD mouse model that was recently published by our group that expresses both APP-PS1 and tau. So we will have the main pathological hallmarks of Alzheimer's disease. In those animals were treated either with vehicle or the CT1812 drug. The groups were balanced equally for sex, that will be important, as you will see. And we collected blood and brain drug concentrations, and we performed a similar study that we have done in humans using array tomography and FRED in the entorhinal cortex of those mouse. So what we found when we treat those animals, so first of all, just globally, I want to show you that if we compare the control and the APP-PS1 tau mice, and we quantify the synaptic density, we see a reduction of synapses, of postsynaptic terminals in the treated mouse, and an increase of A beta and an increase of tau, as expected in this model. However, we could not see differences between the vehicle that you can see in green and the treated mouse that you can see in orange. In all the pathologies, nor the synaptic density. We didn't find differences also regarding the plaque distance, not the synaptic localization of its amyloid. Interestingly, what we did see is that if we put together the treated animals, we can see that if we look to the receptor occupancy of the drug, the percent, we see that in male mice, this is much higher, is statistically significantly higher than in female mice. Even more, I said in the beginning that his colleagues found a cognitive improvement of mice treated with a sigma-2 receptor antagonist, and this was happening when the receptor occupancy was above the 80%. So that's why we next wanted to study what was happening in these APPPS1 tau mice that accomplish this condition, so that we have a therapeutic window. So again, here you have a similar stain that we have seen in human cases for the vehicle and treated groups. We could not appreciate the difference at this point. And if we go to the densities, again, we don't see differences. Here, again, we are just showing those animals that have more than 80% receptor occupancy. We found no differences in the localization, in synaptic localization of A beta or TMEM97, or both TMEM97 and A beta. And we found no difference in the percent of synaptic threat. So it seems that we are not stopping this potential interaction. Also, if we look again now to only to this APPPS1, we see this increasing receptor occupancy in the male mice that is quite interesting. The only significant result that we found is that if we look to the correlations between the different measures that we investigated, here I have highlighted in the black box those significant correlations, and we found, as expected, that as more beta amyloid we have, as more synaptic terminals beta amyloid, as more TMEM97, as more synaptic terminals TMEM97. But what is interesting is that we saw that as more receptor occupancy, as less Fred positive pixels we found. That is this negative correlation that is shown here. However, it's just 5 cases, those 5 mice APPPS1 tau that had a receptor occupancy over the 80%. That is what it has been described to be this therapeutic window. So in summary, taking together, what we have seen is that in human cases, in 80 cases, there is an increase of TMEM97 total levels, as well as synaptic localization of it that is not related with the plaque proximity. We've seen also that TMEM97 can be found together with beta amyloid close enough to generate this Fred effect, which may suggest interaction. And this is supported also by the observation of other proteins. For example, the prion protein, the cellular prion protein with beta amyloid. In the mouse model of AD, we saw a reduction of amyloid beta TMEM97 Fred potential interaction in relation with the receptor occupancy of the drug, but the treatment with a TMEM97 antagonist does not decrease the total levels of TMEM97 nor the synaptic recovery. Finally, I just want to acknowledge special thanks to the donors and their families, and to the lab members of Prof. Jones and Jones. And thank you very much for watching this video.